Good afternoon, everyone. Today we will be presenting microbial growth puddle water study versus dirt. I am Megan Turner.
My name is Whitley Reynolds.
I am Jean-Pierre Adelon.
I am Piper Lyons.
And I am Kennedy Hurley. For our information and background information, we'll start with how the environment influences microbial growth. Microbial communities are shaped by their environment. The moisture, temperature, nutrients, oxygen, and pH determine how microbials can grow. Even small changes like rainfall, soil moisture, or oxygen can affect the nutrient diffusion and microbial survival. Ideal environments for yeast are, I want to say they thrive more in nutrient rich environments like fruit surfaces, decomposing plants, sugary materials, and in soil. And some of the ideal environments for bacteria are found in soil, water, air, and living organisms. But some species do differ in oxygen preference and pH tolerance, leading to different growth patterns in dry dirt versus puddle water. And then we have why studying microbes is different from environments. We have why studying microbes from different environments matters. Isolating and studying microbes lets us see how environmental factors can affect growth, diversity, and physiology. Observing colony morphology, things like gram reaction, pH, and metabolic activity show how microbes can adapt to their habitats.
For our hypothesis, we said that microorganisms are present for the dirt sample. For environment A, our dirt sample was collected from a parking lot. And as you can see in the left picture below, that is where we collected the sample. For environment B, we collected the puddle of water sample near the building, and you can see that in the picture on the right.
And so for this experiment, we isolated microbes from two different environments, sidewalk dirt and puddle water. We collected every sample aseptically and plated them onto nutrient agar, then incubated the plates at 23 degrees Celsius to allow colonies to grow. And then after incubation, we analyzed the bacteria using colony morphology, gram staining, and fermentation testing. We specifically chose sidewalk dirt and puddle water because they let us compare two different habitats, a dry area with limited moisture and nutrients versus a wet environment that naturally supports more microbial life. The sidewalk dirt is exposed and dry, which restricts bacterial growth, while the puddle water contains moisture and organic material, making it ideal for a more diverse microbial community. Comparing the two helped us see how environmental conditions influenced microbial diversity.
So for the results, it shows the overall bacteria growth trends. The pie chart in the graph illustrates how the bacteria increased over time and how different variables influence the rate from the growth. From this, we can see a clear pattern. Certain environments supported rapid growth while others slowed it down significantly. This provided our first indication that environmental factors play a role in bacteria activity. So you can see on the Agna plates is used in the experiment. The first plate shows noticeable more bacterial colonies compared to the second. This visual comparison highlights the effect of independent variable, whether that is temperature, moisture, or another factor that is tested. The difference between the plates demonstrated how even small environments can drastically change colony formation. So this contains the pH testing results. These strips show how the bacteria samples altered the pH of their environment. This is important because bacteria often change the acidity of the medium they grow in. The color shifts on the pH strips confirms that growth was occurred and also help us determine samples were most active metabolically. In the last slide, the image displays clear differences between the contamination level before and after the experiment. The bright areas on the scan represent bacteria presence, and the changes between two images can help validate the growth. The point, the dirt is the dirt. The dirt. I'm sorry, gram positive, and the dirt sample, the water sample is gram negative.
For our discussion and conclusion, our hypothesis was accepted. We hypothesized that microorganisms are present in the dirt sample. For the dirt sample, the pH level of 5 was concluded and it did not fermentate. No evidence of fermentation was on the tube after incubation. This suggested the presence of non-fermenting or slow fermenting soil bacteria, and there were also rod-shaped bacteria of this sample. For the puddle water sample, there was a pH of 6, and this was the only sample that had evidence of fermentation. The bubbles indicated fermentation, and there are clustered cockeye-shaped bacteria for this sample. Potential areas included contamination during handling and temperature changes during transport. Future improvements include checking plates daily out of the 5 days of incubation, using replicate plates, and incubating samples at a controlled temperature.
For the discussion and conclusion continued, this experiment is relevant because it allowed us to identify microorganisms in soil and water, which helps us understand the importance of environmental health, nutrient cycles as well as ecosystem balance. The tests that we use were gram staining and fermentation, and these same methods can be applied in the real world, allowing us to detect contamination and identify potentially harmful bacteria. We modeled this and allowed for scientists to monitor microbial diversity and protect both the ecosystems and public health.
This will conclude our presentation. Thank you.